We made it to the reproductive system, and we'll have a quick intro and then dive into the male reproductive system, looking at the scrotum and the testes. And there's surprisingly a lot to know about the scrotum. So, the functions of the reproductive system, well, really, this is all about creating offspring. And we need to have gonads to make the gametes so that those can make the offspring. And then along the way, we'll have these sex hormones being made and these secondary sex characteristics we'll talk about. But those also go into making sure we can create offspring. So that's the real function of the reproductive system. So when I say we need to maintain the gonads, we need to know that the gonads are reproductive organs that make gametes. These reproductive organs are testes and ovaries. Both of those are gonads. It's not just males that have gonads. Testes and ovaries are both gonads. And they make gametes, which are haploid cells, meaning they have half the amount of chromosomes needed to make a human. And those will combine to make zygotes. So male gametes are called sperm, female gametes are called ova, which are the eggs. You take one sperm, one ovum, one egg, you combine it, you get a zygote. So what is an embryo, and what's a fetus? Good question. Let's create some offspring. We combine these two gametes, and we form a zygote. So that's a single cell that's formed when the sperm and the ovum combine. And then that one cell will grow and then divide into two cells. And those will grow and divide into 4 and 8 and 16 and 32 and 64 and 128 and 256 and 512 and 1024. And rapidly, that ball of cells grows and grows and grows. Around 5-ish weeks, the name changes to embryo. And it's around this time that those cells actually begin to form tissues and organs. In fact, as early as like 5 to 6 weeks, but usually a little bit later, you can start to hear the heartbeat of the embryo. So clearly, organs are being made at this time. So what is a fetus? Around 11-ish weeks. Well, that's when you start to have organ systems. Then that heart is not only beating, but it can start to circulate blood. The other thing that the reproductive system will do is synthesize sex hormones, which contribute to secondary sex characteristics. So first, what are these hormones? Well, testosterone is produced by the testes in great amounts. And the female hormones estrogen and progesterone are also created by the ovaries. And the biochemical pathways of this are kind of complex because some of these are precursors to other hormones, something you probably get into during physiology. What we want to understand is that these sex hormones will facilitate the reproductive processes. As one example, the ovary and the uterus need to be in communication so that when an egg is sent on its way to the uterus is prepared. So that's one function of these sex hormones, is to facilitate these reproductive processes. And the other function is the creation of these secondary sex characteristics, which are characteristics that arise after birth, right? So if you have a XY human baby born and it has a penis and testicles, testes, then those are characteristics that are present at birth. But if you wait long enough, that XY baby might have a beard, increased muscle mass, enlarged thyroid cartilage, which lowers the voice, pubic hair, etc. So these are secondary sex characteristics. They become present later on. See if you can also name some secondary female sex characteristics. And if not, you can always look it up. You might find something surprising, like I think my wife's arms are pretty long. I don't know if they're visibly longer, but they're long. But in fact, all females have slightly longer arms when compared to males of the same height. It's just one of those things. Okay, so that's just some basic terminology. What's an embryo? What's a gamete? Now we can get into the male reproductive system, and we'll start with the scrotum. It seems like it's just this sac that hangs there underneath the abdominal pelvic cavity, and it is that, but that and more. So it's divided into two scrotal cavities with this septum in between. Remember, a septum is a wall. And each cavity has one testis, and so plural we call them testes. But the big thing about the scrotum is that it's providing thermoregulation for the sperm. Because sperm are sensitive to these higher temperatures of our core body temperature, that 98.6 degrees Fahrenheit. They actually need to be kept in cold storage. It's not super cold, but we're talking about 95 degrees Fahrenheit. If you warm those puppies up, they get real active. They'll be trying to swim and all this. And that's good if they're warmed up inside the female anatomy and they're trying to swim to the uterus. But in males, if they're kept too warm, like even highly constrictive underwear can do this, they're damaged. They won't be able to properly grow and mature and survive. So you need to keep them a little cooler. How is the scrotum contributing to this thermoregulation? Well, it does so in three ways. Something called the pampiniform plexus, then the cremaster muscle, and dartos muscle. So let's look at these structures and how they keep the sperm cool. Okay, this pampiniform plexus, which means formed like or shaped like a thread or a string or something, is a network of veins that cool down hot arterial blood. That's a little complex, so we'll look at that in a second. Let's look at these other two first. The dartos muscle decreases or increases the scrotal surface area. So just like this jackrabbit, if it's real hot in a desert environment, it can lift up its ears and it will increase its surface area and help it cool off. If it's cold, it can lie its ears back and it gets smaller. It decreases its surface area to stay warm. Same way, if you're really hot, you might spread out on the couch or on the floor, and if you're cold, you might curl up tight. So the scrotum spreads out on the floor or curls up tight if it wants to release heat or save heat. And that's due to constriction or relaxation of the dartos muscle. Surprisingly, each testis can also move up or down separate from the other testis. And that's due to the cremaster muscle, which are like these little elevators that can elevate or depress individual testes. This is generally smooth muscle. It's not voluntary, although there are some like Kegels and stuff you can do to try to replicate this. But in general, not something you need to think about. So let's go back and look at this pampiniform plexus and figure out why I have this picture of a penguin. Well, here's a duck, which is the best I could do. Imagine a duck on an icy pond. And this duck has this cold, freezing cold blood making its way back up its leg. Now, you really do not want that blood to go back and hit the heart. Like, you'll go into shock from this frigid blood coursing through your body or the duck's body. So, what are we going to do about this freezing cold blood? Well, there's still another problem, actually. The duck is pumping this super hot blood down into its feet. And meanwhile, it's giving up, or could be giving up, all this heat, all this body heat, into this icy pond. So this duck is going to burn through all its energy reserves and die. So, what to be done? Well, when you have that hot blood coming down to its cold little feet, that artery must travel through a plexus, an interwoven network of veins, every which way. Not just one vein, but a whole interwoven network, a plexus of veins. So as this artery comes down, rather than give out heat to the environment, what happens is it heats up this cold, cold blood that's going back to the heart. Okay? So we've solved both problems. The blood that's going to reach the duck's body and heart is relatively warm now. And the blood that's going down into the icy cold feet has given up its heat, but not to the frozen pond. It's given its heat up to this network of vessels. That's what happens here. We have a testicular artery, which is this one, and that's this super hot blood that's going to come down to the testis. But we can't have it do that. It's going to cook the sperm, figuratively speaking. So that testis has to go through this network of blood vessels, the PP, pampiniform plexus, and it will get cooled off before it reaches the testis. Just a few degrees, just enough to make sure the testes are not overheated. So I'll go back. That is why our testes are like that. Same reason that bird feet and duck feet have the same system. You saw the dartos muscle and cremaster muscle. And here's the cremaster muscle, remember, just on one testis, because these muscles can contract individually, whereas the dartos muscle is in the wall of the scrotum, causing it to wrinkle, to warm up the testes, or relax to loosen the scrotum and get the testes further away from the abdominal pelvic area to cool off. Finish up with the scrotum, let's recall that there are two scrotal cavities with the septum in between. And we talked about the pampiniform plexus and that testicular artery and how they come down and service the testis like this. But anytime you have blood vessels, you also have lymphatics coming down. And of course, there's nervous tissue to monitor what's going on in the testes. And there's also the tube that the sperm will eventually use to exit this area called the vas deferens. And so what you should be seeing here is that there's a whole bunch of different structures and they're all bundled up together. And we have a name for that bundle. It's called the spermatic cord. So it's a sheath that maintains or captures all of those different vessels, etc. And the last thing to know about the scrotum is that it contains the testes. And if you're to look at a single testis, one thing of note would be, how did the testis get there? Because actually, lots of mammals have abdominal testes that live safely tucked inside the abdomen. So they have to have other ways to keep their testes cool in some cases. But our testes have to exit the abdomen to reach the scrotum. Usually that happens just prior to birth. So in the next slide, we'll see a little opening that the testes use to drop into the scrotum. And we'll finish with thinking about the tunica vaginalis and tunica albuginea. These are basically coats. So tunic means coat. And the vaginalis means sheath coat. In fact, vagina means sheath, which means that some presumably male person named the vagina in the context of the penis. You know what I mean? reproductively speaking, that's the point of it, is procreation of the species through penis and vagina. But strange. So this tunica vaginalis means the sheath coat, and it is a serous membrane. Remember, double layered membranes, little cavity in between with fluid, and it's actually part of the peritoneum that tucks way, way down into the scrotum, helps protect the testes. The tunica albuginea, the white coat, is kind of more like a capsule right on top of the testis. So let's look at this stuff. Really, we're just trying to finish a few things here, like find the spermatocord, look at all this stuff in there that's all captured within the spermatocord. Find the inguinal canal, kind of runs up like this. And this ring at the bottom is called the inguinal ring of the inguinal canal. But it's through here that the testes drop down. It's also through here that you could have some of your intestines drop down into your scrotum if you have an inguinal hernia. And last, we can see a testis here with the tunica vaginalis. In fact, 2 layers, since it is a double-layered serous membrane. So let's look at these testes more closely. Here we just see the tunica albuginea, this white coat, but look how it subdivides the testis into these little lobules. So these are called septa. You know, a septum is a wall. So these septa divide up the testis. And inside each of these lobules are several seminiferous tubules. We'll talk about these on the next slide, but one thing they do is make sperm. If you track a seminiferous tubule, let's take a closer look. The seminiferous tubules make this coiled pattern, and eventually they start to straighten out. And we call those straight tubules. And then they form this big plexus-looking structure called the rete testis. Fun name, important job. It's basically an anastomosis. It allows the sperm to have lots of alternate pathways it can use to exit the testis, which finally it will do through the efferent ductules. Remember, these term efferent is always referring to an exit. So here, the sperm are exiting the testes through the efferent ductules and land in the epididymis. If we take a closer look at the seminiferous tubules, like through a cross-section of a testis, what you'll see are a bunch of round or round-ish tubes, and these are all just cuts through the seminiferous tubule. What's happening here is that the very outside of the tube has some stem cells that will become sperm. And over time, those cells divide and change as they head towards the lumen. And by the time you have a sperm that's finally all made and packaged up and ready to go, it falls into the lumen and gets wished off to the epididymis. A little more specifically, so we start with, we said stem cells. The name of those stem cells is spermatogonia. And they will begin the process of spermatogenesis, the creation of sperm cells. So where do we find spermatogonia? On the very outskirts of the tube. All these cells here in the very outskirts are spermatogonia. Then those will turn into spermatocytes, which are found all through the kind of central region of the seminiferous tubule. Those will undergo meiosis, so a special type of cell division. And you will have 4 unique spermatids clustered right around the edge of the lumen. Once those spermatids are ready to go, they kind of fall into the lumen and get washed to the epididymis. And I'm being careful with my terminology here because I want to make sure I'm not implying that they jump off and swim to the epididymis, because these are still immature sperm. They still need to go to the epididymis and mature for a while before they're ready to swim. Let's run through this process of spermatogenesis one more time. Imagine that this is one cross section of a seminiferous tubule, and on the outskirts is where you'd find your stem cells, spermatogonia. When they divide, one spermatogonium will stay behind, and one we will call a spermatocyte, and it will start moving towards the lumen. That spermatocyte will divide and form two spermatocytes, and then those will divide. Now you'll have actually 4 haploid cells. Each of these now only have half what they need to really be a fully living cell. But they're still these big, round, dorky cells. They're not ready to meet with an egg. What has to happen is these big cells have to thin down and grow tails, flagella. Now we've got stuff that looks more like sperm because these are spermatids, which are very close to becoming mature sperm. They've got their tails, they've lost a lot of organelles, they've lost a lot of cytoplasm, so they can focus just on their one job, delivering their DNA to an egg. And then once these fall off into the lumen, now we have, these are actually sperm. So if you were to look at the histology of a testis, I wouldn't be too concerned about telling all these cells apart. I would just say the outer edge, all of those are spermatogonia. The very inner layer of cells are spermatids, and everything in between are spermatocytes. So, what becomes of these sperm cells after they fall into the lumen and drift away? I'll tell you in a second. There are also 2 other types of cells in the testes. Interstitial cells, that basically means the in-between cells. In other words, if you look at a testis in cross-section, there's a whole bunch of cuts through the seminiferous tubule. But then there's all this in-between space. So all of these spaces are where you would look for interstitial cells, and their job is to make testosterone. And finally, sprinkled amongst these other cells are nurse cells. Nurse cells have a few jobs. They're also called sustentacular cells, which is kind of good because it reminds us they have something to do with providing sustenance. Or they're called Sertoli cells after the dude that discovered them. And they're supporting this process of spermatogenesis. So they are nurturing the developing sperm cells. They maintain a blood testis barrier, basically providing a very tightly controlled environment for the sperm to develop within. And if sperm count is too high, they'll make a hormone called inhibin to inhibit the production of more sperm. So, next time we will follow the sperm as they mature in the epididymis and eventually move on through a variety of tubes, mix with a variety of substances to form semen, and then visit the female anatomy.